All right, so welcome everyone to the final talk of our micro course here at Emory, going over how to use Fiji in bioimage analysis. Today, I'll be covering colocalization with all of you. So most of you have been previous attendants, so we will go ahead through all of the housekeeping. Remember, if anyone has questions, they can schedule a consult with us. We're always here to help our fellow researchers and colleagues. But what should you know by the end of this course, or have a better understanding of if you already have some background in colocalizations? You should understand fundamental concepts and more especially how to apply them. We'll go over a number of coefficients, all of which tend to add something to your understanding of how the data is interpreted, how the components interact with each other, none of which is useful really by itself, absent a deeper understanding of its function and utility. And finally, after we cover the basics of colocalization, we're going to go through a few examples of using the easy colocalization plugin for Fiji. It was included in the material for the course today. If any of you had issues installing it, we'll cover that as well when we get to that point. But it is a very versatile plugin and very good for this kind of work because it produces all of the statistics we'll talk about today. So first, what is colocalization? Many of you might have heard the term. For those of you with less experience in it, it's the study of the spatial overlap of two or more fluorescent labels in a sample that you have. Colocalization implies a relationship between labeled compounds, but it doesn't actually directly prove most of those relationships. And we'll get into why that's the case because of the techniques that are used. There are limits. There are resolution limits that play an effect in how data should be interpreted, and even when colocalization is applicable. And to mention as well that colocalization doesn't necessarily mean that two objects are right on top of each other or even attached to each other. It merely provides circumstantial evidence. For example, if you were to think about it as people inside of a building, colocalization could tell you two people are inside the same building. It couldn't tell you that those two people are in the middle of a conversation or even in the same room. But it can give you the circumstantial evidence to say that these people are in the same building. Let's interrogate this sample or this system in a different way to figure out if they are in the same room, if they are interacting with each other. And for questions like that, techniques like Fret, Foster, resonance energy transfer is a good avenue to continue the research, because those kinds of techniques, which rely strictly and directly on the interactions of two very small components, would then provide evidence for their direct interaction instead of co-localization, which just indicates that they're in the same region. So we went over the very basics of what co-localization is, but what are the resolution limits, as I mentioned? So here you'll see a diagram of an airy disk, which is light coming from a point specimen and passing through the objective and reconstituted as an image. The amount of resolution we're able to get on an object or a structure relates to the wavelength of the light used and the nature of the objective we're using, the aperture in general. So smaller wavelengths of light are generally able to resolve more detail, but they're also more harmful to a lot of samples. Longer wavelengths, can't resolve as fine, can't resolve details as finely, but they're also less damaging to tissue. For microscopes, typical optical microscopes, I should say, we can achieve a resolution of around 250 nanometers. There are more intricate techniques, such as STED, some of you may have heard, that are able to get even smaller resolutions. But we won't talk about those today. Rather, we'll just limit ourselves to conventional optical microscopes with a maximum of, say, 250 nanometer resolution. So that's very small, but still it's much larger than some structures like proteins that we might be interested in looking at. The diagram here shows you that kind of diffraction. So part A is just a single point source. We're looking at it through an objective, and we see how that detail is kind of diffused slightly. In instances where two objects which undergo this kind of blurring as a result of just the physical parameters of the system, when they are very close to each other, like in C, we have trouble resolving that. But it's also one of the interactions that co-localization benefits from. If two labeled components are close enough together that they can be imaged in one wavelength and in another, you would get those pixels bleeding into each other, and you would say that something is co-localized. But is that always appropriate? Some very small structures, as I mentioned, can't be resolved past our lower limit of 250 nanometers. So even if the structure is smaller than that, it will appear that size in the images we take, which, to go back to the building analogy, can be troublesome if the thing we're looking for is very small and the area it can occupy is very large in cases like this. So let's show the example here. If the true object is larger than our diffraction limit, or the size of our diffraction limit, then the object we image, we can be relatively certain, is of a similar size as it is in real life if we set up the experiment correctly, set up the microscope correctly, and look at the data appropriately, all of which depends on the project you're working on. But if the true object is very, very small, we would still image this blurry, very large version of it in an image. And if those two, say we wanted to compare it to co-localize those two signals, and the structures themselves are very small, we are less certain. The example I'm showing here is if we had two very, very small, let's say 25 nanometer objects that we've labeled, and we want to prove that they interact with each other, we reach a limit to the utility of co-localization because those two very small objects will appear much larger when we capture that image. And it will seem like they're co-localized even when, in this example specifically, they're separated by some kind of barrier. So these two objects are not in the same room, they're not talking to each other, but because of the limits of our optical microscope, they may seem like they're in the same room, that they're very, very close to each other, even when we can't really be sure that they are. So this is kind of the limit of co-localization. it's good for larger objects like cells. It is less good for things like proteins, in which case techniques like FRET would be a lot more useful in actually proving an interaction instead of just suspecting an interaction. Up to this point, does anyone have any questions, any confusions? Gaurav, do we see anything?
No, all good.
So I
No.
just have a question.
Yeah,
Like
go ahead.
normally we do want to see the protein co-localization, right? Protein-protein interaction.
Mhm.
So you're saying it's difficult to see those interactions.
So it's circumstantial. You can localize a protein to a certain part of the cell and say this is where it goes, and you can say that relatively confidently. But if both of your objects are very small, it's harder, almost impossible, just with co-localization to then say these two very small objects are co-localized in our data, therefore they're interacting. And most you can say is they're in a similar region, but to prove that they interact with each other directly, you'd need a little bit more. You'd need something like FRET. Does that make sense?
Okay. Because earlier we just said Pearson's coefficient or something, and then it said, yeah, these two are co-localized.
Yes.
Yeah. So the like just giving that is not sufficient, you're saying.
Exactly.
Pearson's coefficient. Yeah. Okay.
Yeah.
And if I may add one more thing to that, it would be, let's say, if you have one protein, for example, the green in color is cytoplasmic, and on top of that, you have lysosomes, which are part of the cytoplasm. And if you were to run a colocalization analysis, of course, your lysosome is part of cytoplasm, but that doesn't mean they're co-localized because they're completely two different compartments. So that's another thing to think about when you talk about colocalization versus what is the physical importance or physiological importance of colocalization.
Thank you, Gaurav. That's a good point.
Yeah, thank you.
Okay. This discussion brings us into a quick note to LUTs, which is lookup tables, which is how you give your grayscale data in apparent color if you're looking at it. To anyone who attended the micro talk on data analysis that I gave a few months back now, we went over that there. For anyone else, the human eye, and this kind of tangent will make a little more sense once it's finished. The human eye is good at seeing some patterns. It's better at seeing some colors than other colors. So it's important to consider a lookup table when you're just looking at data visually to get a sense of what it's like. So these 4 images here, they're all of the same data set, but they use different lookup tables. Already you can tell some of them are much harder to visualize for us than others. And that brings up the consideration of what is a good lookup table for me to use when I'm trying to communicate with my data. And that depends on what you're trying to show, but a good rule of thumb is to utilize cyan, magenta, and yellow, and black, because those are actually colors a lot of people can identify. And if you note the link on this slide, there's in fact a colorblind toolbar for Fiji to help you use the best kind of lookup tables for your data. And that colorblind bar will help make sure that you're using colors that even people with color blindness are able to see effectively. And it's a benefit even to people who don't have color blindness, because we can compare the cyan and the blue representation of the data, and we see cyan a little better than we see blue. So those fine details we would see in a cyan LUT, we might miss in just a blue LUT. But why do we care about that? Well, it's important because when we are analyzing data or we're looking at initial data for co-localization, relying just on the color things are and how well we interpret that is limiting, and it can be dangerous in times. That being covered, just a quick note, any image you see is data. It's an array of pixel values, and the way that the image is stored by the computer is a pixel location and an intensity value for how much signal that pixel has. That's what data is. When you look at any kind of image, you're seeing gray scale values, intensities that have been partnered with an LUT. Grayscale values are just those black and white intensities. So when I use terms like for every pixel, that is for an image, every pixel in the array that comprises that image, we might do a certain calculation or a certain analysis of that. The overlapping of two images as well. I mentioned we want to be cautious when we're analyzing images just by eye, because our eyes did not evolve to perform fluorescent microscopy. One of the common ways that people sometimes investigate for co-localization is to just take two images, put one on top of the other, and see which pixels change color. This can be useful as a first order analysis, like, hey, yeah, there looks to be some co-localization in this set, but it can be misleading and it can be dangerous. So the reason I mentioned the LUTs and what an image is for the examples right here. We've got the same data sets and we're overlapping the image on the left with the image to the right of it. And we say, wherever the color changes might be co-localization. So this is interesting. So we'll look into more detail, but how apparent that overlap is very dependent on the LUT you're using. The examples here show a bunch of composite images. Not all of them clearly indicate, to me at least, which sets are overlapping with others, and then what that kind of means in terms of relative brightness, how strong the correlation is, how strong the co-localization is just from that. So it's something to be mindful of when you're using these in your analysis or in your manuscripts. Because, for example, from the top and the bottom, between those two, they give different impressions of which components are co-localized, which colors are changing, and how the colors are even changing. And that is not, in fact, the only consideration. The intensities for the different pixels and how those images are then shown on a computer screen, for example, can have an effect on the apparent colors. So what we've seen before is what those images look like to us by eye on a computer screen. That doesn't necessarily mean we know what the data itself looks like. The data as we discussed on this slide, is an array of pixels, each with intensity values. But your computer screen may not always show that. Something that can happen when you go from data to visualizing it on a computer screen, for example, is depending on the bit depth of the image, that is how many potential intensity values a pixel can represent, can get washed out. If you have depending on the computer screen, you can have a process like the one illustrated below occur, where you have a red pixel and an orange pixel in the data file that are near each other. But your computer screen can't project something with such fine granularity, let's say. So instead it says those 4 pixels, we can't show that, we can show 1 instead. So we'll take an average of the colors there, that's what we'll project. So the kind of overlap that could indicate co-localization is at once also subject to the medium through which you're seeing it. If you're looking at it on a computer screen, it could seem like there's co-localization when in fact the pixels are not on top of each other and would not show up as co-localized. Does that make sense to people? Do you have any questions about that? It's a very key consideration going forward. Okay. Now, after all those considerations, we'll get into some of the actual coefficients that we'll use, and these are going to be determined mathematically as we're looking at the data. So some of the ones that we'll focus on are the Manders overlap coefficient, Pearson's correlation coefficient, and its kind of modification for nonlinear relationships, which is Spearman's rank coefficient. And then we'll also briefly go over Costas randomization, which is a way of statistically making sure that something which looks like correlation is a statistically significant correlation instead of potentially being the result of just accident or noise in the system. There is another very common way to look at data you're interested in determining if it's co-localized, and that is an intensity scatter plot, where for 2 images, you'd have all the pixels on one image with their intensities matched against all of the pixels of the other image with their intensities. And the results can vary. What the examples here are of one set of data that has actual co-localization in it, and then another set that doesn't have any co-localization. So we can see for a set that is co-localized, you would expect some relationship between the intensities in one channel of data and the other channel. For non-co-localized sources, you might see something like in the second plot. And when we say, or when I say rather, when these 2 pixels are compared by intensity, we're always saying that for each image, pixel by pixel, it's compared position-wise, which means if we go back here, you have, say, 2 sets of data that you want to check for co-localization. Here, one is essentially you would you would start to compare the two pixel by pixel from start in the same way for each image. Does that make sense to people? Does anyone have questions about that? Okay. So first, let's start with Mander's coefficient, co-occurrence. And this is here just to illustrate the mathematics that go into it. You're not going to need to do any of this yourself. When we get to the easy co-localization plugin, I'll show you how to get all of these coefficients. But for those who are interested, here is the mathematics of it as well. So Manders, what it measures is essentially the overlap between two sets of images, the intersection of a set of images with different labels. So you can see here, we've got a red signal and a green signal, and we are determining the Manders coefficient for that. How much of the red signal overlaps with the area where we get green signal, and we can calculate that for red into green and for green into red as well. As I had mentioned before, this works very nicely for larger structures, less so for structures which are sub-resolution, like proteins, viruses, or even substructures of a cell. This coefficient is very resistant to changes in the signal-to-noise ratio specifically, which means the MOC here is the Manders coefficient we're looking for. We can see that even as the SNR gets lower, so we have more noise to signal, it remains very close to itself for these different signal-to-noise ratios. We can see here at the top that to be the case. So we have noisier data at SNR equals 8, but the MOC remains close to what it was with a higher SNR. What it can be very problematic is because this coefficient takes into account not only the position of the signals, but also the intensity of them in terms of the overlap. Any kind of background noise or additional brightness will skew this coefficient in a way you don't necessarily want. So the example at the bottom here is that if we have background signal that we fail to remove from the final image, that can skew this coefficient that we're looking at into being higher, even if it doesn't, even if that higher coefficient doesn't then indicate that the two structures or two labels of interest are more co-occurring.
So Stoyan, it's a great point to make over here, which is if anybody's goal is to in fact measure co-occurrence or co-localization using Manders coefficient, and they are imaging using a confocal microscope, they should really make sure that they use the feature of linear averaging or the line averaging, so that all this pixelation that you are seeing with low signal to noise ratio gets averaged out, and you get something which looks as the signal, which is part of signal to noise ratio of 19, and that will increase your Manders coefficient.
Mm-hmm. That's a good point. Yeah. That's something to keep in mind, that this coefficient is resistant to changes in the SNR, but is very susceptible to changes introduced via background noise, for example. Another component of making sure you're getting a Manders coefficient and then interpreting it meaningfully is how much of this co-occurrence may be due to something that can occur randomly. So how would we look at that? We've got here the examples A and B. As an original image and then as a randomly modified image. Now, the Costas method takes a look at the whole image and then modifies pixels in a way. It moves them around, and then it performs the same set of calculations to calculate Manders coefficient. And the idea is, if this is a co-occurrence that can occur randomly just as a function of noise, then every time we modify this image and run the Manders coefficient calculations on it, we could get something that is similar to what we did with the two initial images. Now, the way we are interpreting the histograms at the end here at the MOC is for a lot of variations, and for every variations we calculate Manders coefficient. When we plot those in a histogram, what is the distribution? The distribution of A indicates that as we randomly change one of the images and then calculate Manders coefficient for that we see a lot of variation, and then the preponderance or the variation in the MOC drops off. And then the one we're looking at, which is here with a red point here, that is the one that we calculated initially. And how does its value compare to all the other values we calculated after we randomly modified one of the images? Well, most of them fall away from it, below it. And we see that, and the conclusion we can draw from that rather is that for the example A, it is very unlikely that our calculation and our coefficient is the result of just random chance. So it's like for a p-value of 5%, our MOC is accurate in the case of A. In the case of B, however, we have the same coefficient calculated. It's also indicated by that red arrow, but because of the way the data looks, it is a calculation that could occur randomly pretty often. So it's not a coefficient for example B specifically that we can reliably say this did not occur by chance. Now, does that statement make sense to people? Any questions? Okay. This method is very computationally intensive as well. There's a way to do it with easy co-localization. the plugin we'll get to later, but it is useful to have, to utilize in determining Mander's coefficient specifically, whether it is genuine signal or something that could happen randomly. For reference, a summary. Mander's coefficient is not very sensitive to SNR, but it is very sensitive to background noise and noise in terms of the intensity, pixel intensity. Now, let's move on to Pearson's coefficient, and that basically determines to what extent variations in intensity from one image can be described as a function of variations of the intensity in another image. This coefficient, though, works for linear relationships, and we'll get into a few examples later on where that linear relationship is clear what it means. If we go back here, for example, the co-localized signal on the left is linear-ish. Like you can draw a line through that data with a linear regression. That is, that's a kind of signal that could be accurately described by a Pearson's coefficient. Now the variation for this is between -1 and 1. And here are some examples of the kinds of scatter associations and the kinds of values that the Pearson's coefficient can have. Now at the bottom, we can see a lot of examples where the Pearson's coefficient is 0, but it's 0 for a set of data that can be very well, if non-linearly, behaved. So here for that signal that looks like a W, there is a correlation in the intensities of one image to the intensity of the other. It's just not linear, and so the coefficient here indicates will be 0 even when there is a link, even when this behavior in the intensities of one image is relatable to that in the other. Does that make sense to everybody? Okay. We'll also make a point here of going over the fact that Pearson's coefficient, the way it's calculated, is in fact sensitive to the signal to noise ratio. So the lower that value becomes, the lower the Pearson's coefficient will become, even if we're not actually changing the relationship between the two things which are labeled. So we see the lower the SRN, the noisier the scatter plots get, and also the lower the Pearson's coefficient becomes as a result of that. Even if the relationship in the data remains the same, or the relationship between the two components remains the same. Now the Spearman's rank coefficient here attempts to correct for that lack or that failing in the Pearson's coefficient. It attempts to linearize those behaviors so that at the end you can linearize a nonlinear behavior and then come up with a coefficient for that and see how strong that correlation is. For example, here, B and C, in the top, we have a linear relationship, and our rank correlation coefficient and our Pearson's coefficient are very similar. For C, where the relation is still correlated but not linear, you can see the PCC is lower than for B. But if it's treated with Pearson's rank correlation, we can get a better SRCC. And finally, can we have correlation without co-occurrence? And the answer is yes. We can have high co-occurrence but low correlation, like in A, and we can determine that the correlation is low because the intensity plot is all over the place, even as in the image, we can see that there's a high tendency for co-occurrence. And in C, we can see that even though there is low co-occurrence, there's still a high correlation in the, say, pixel intensity plots. So yes, both of these things can occur independent of each other, or they can occur together, which is why it's important to look at all of the coefficients that we've discussed and know the data that you're looking at, be able to understand the interactions.
So Stoyan,
Yeah.
for A and C, does this depend on the area within the image that you are using to analyze this? So for example, in C, instead of the entire area that is projected in the image, if I was to pick one specific bead where both red and green are co-occurring, would that give me both high co-occurrence and high correlation if I was to crop one of the bead and then demonstrate that?
That is a good question. And yes, the area you're looking at does affect all of these coefficients. And I will show you an example later on as we're using the Is Co-Localization plugin of instances where defining your region of interest differently can affect the values of all these coefficients. Now we can move on to that. But let me ask everyone, has everyone been able to install the Is Co-Localization plugin that was included with the data for today's talk? Has anyone not been able to install that plugin? Okay. Let us, however, take a quick break before we move into the examples for about 5 minutes. And we will get back to that. So I'll pause the recording for about, all right. So let me show everyone, just in case, how you would install a plugin with Fiji. So if we have Fiji opened here, we can go to plugins and install, and then we can navigate to the location of our plugin. For example, there was an Easy Co-Localization _.jar plugin that was included in the documents for today. We can then select that when we've gone to Fiji's plugins install plugin and select it there. It will show us then the folder, the plugins folder for Fiji. And that folder contains all of the plugins. And it asks us, is this where we want to install that plugin? And we say yes, save it here, because that is where Fiji, that is the folder Fiji looks in to determine what plugins it has access to. So we can click save here. Easy Co-Localization already exists because it was installed before, but we can replace it in, for example, instances where we have a newer version of, say, Easy Co-Localization. We can click yes to replace it there. And then once we restart Fiji, we will see it installed. Here under Easy Co-Localization. So at this point, I can show you an example. Simply output. Now from the data that we've got particles with a, say, a good overlap. And to use this plugin, we can open the data here, good overlap. And under image stacks, we will decompose the stack into 2 images. Now to run the plugin, actually, we can go plugin Easy Co-Localization, and it'll open up this GUI interface. From here, we can select the images we would want to run the colocalization analysis on. That's image 1 and image 2 that we just decomposed from the stack as an example. So that's a good overlap 1 and good overlap 2. The plugin also has a cell identification input. And what that really means is, as Gaurav asked a little bit ago, can we define a region of interest for a set of data that we're looking at, where we don't take into account all of the background, which is black or is not showing us any data? And yes, if we have a set of data with ROI information we want to consider, we can plug that in and specify it here for analysis. If we were interested in creating our own region of interest as well, we would be able to do that as well. So let me, yeah, and we will go through an example of that later on. But for the two images we have, 1 and 2, we will just look at the whole one initially. Under visualizations, we can also specify if we want, say, a cell intensity scatter plot generated, which we do in this instance. And under analysis, we can go through the different coefficients that we want to take a look at. So we will select PCC, SRCC, and MCC, which are Manders, Spearsman's, and Pearson's coefficients. We'll also have it present us with histograms and a summary. When we click Analyze, we'll get all of that out here. We will get a scatter plot. We will get a log summary, which is very useful, a very useful component of this plugin that you would generally want to take a look at, because not only does it contain a breakdown of the various coefficients you asked for, it also provides some detail on how to interpret them, which is very useful, especially as a reference. Here. Yeah. And in fact, we can see for this data here, we get a scatter plot and we can get the coefficients as well. Here. So why don't you all go ahead and run this analysis yourselves with that data. And then see if you get the same values as are listed here. So let's take 15-20 minutes for all of you to play around with that by yourselves and take a look at the different data that was included in the collection here and run that kind of analysis for, say, all of them, the poor overlap, medium overlap, good overlap, and complete overlap sets, and generate these tables of these 4 coefficients. Take 20 minutes to do that, see if you can do that. And let us know if you've got any problems doing that. So I'll pause it.
All right. So so here are the 2 images that we got. And when I do not select any cell identification input, and I go ahead and say analyze. Hold on. I've tried to play with something, and it would be odd if it works out this time versus the other time. See, it's funny, it does work out this time. It did not work out for me last time. What happened? Oh, I know what happened, what I did. I don't know if anybody else had this issue, but I had to disable the measurement, the scaling factor for each of them. So I had to go to analyze, set scale and click to remove scale. If I did not do that, so let me go back to the original data set and try to recreate that problem.
You we're also not seeing your screen. You shared it briefly, but then it went out. Yeah, there it is. Okay. Mhm.
So here I am with both my channels.
Mhm.
So if I do complete overlap 1, complete overlap 2, and none over here.
Mhm.
And after this, if I try to analyze, it says none of the operations is selected. So what I did was I tried to create ROI. So
Mhm.
let's say, for example, I just go ahead and select an ROI over here. That's what I did. And add this ROI to my ROI manager. Now, if I was to select that, and then do analyze. So let me just go ahead and I don't care about that. Visualization, I can enable this, but this is important, as you mentioned. And analysis, let's keep the way they are.
You you actually need to tick which of the coefficients you'd like to capture. So check PCC, SRCC, and MCC. Yeah.
Right. So now if I do analyze, it says scaling factors must be consistent or
Mhm.
global. So I went ahead and I disabled my scaling. I don't know if this is just me who is having these issues or. Okay, so I removed that, and then once I hit analyze, it worked out fine.
Mhm.
But again, this is for one region of interest and not for the entire image. So now if I go back and delete the ROI, and try to analyze the same thing for this data set. Let me go back to my inputs,
Mhm.
none. Um, it does work out, right? So I don't know if the limiting factor over here is that set scale, but I still want to try out with the original data set with the scaling and try to see how that behaves. So let's say if I'm here, so now I got 1, 2, none. And I do analyze, none of the operations is selected. I think, hold on, so
Operations are what
the
it calls
visualization. Okay.
the coefficients. Yeah, under analysis.
All right, so this time it accepted. It did not give me issues with the scaling. Okay. All right, cool. Thanks, John.
Yep. Right now, how's everyone else doing? Has anyone run into problems, or does anyone have questions? Good. So what are everyone's values looking like? Does anyone want to share what they've got? So Gaurav did something that I want to go over as well, which was he created a region of interest using the ROI manager. And one of the examples he had. So open. Actually, let's go to mitochondria. Let's open 2 sets there. We'll go into what to consider for your regions of interest and why that could be important in terms of co-localization. So what the region of interest specifies is what part of the image you want to run your analysis on. Now, it's important to consider if you need to include any kind of ROI specification for your analysis. Usually, if your image is not all structures, usually you'd want an ROI, because as you include any of the dark background in an image, in a fluorescent image or a set of images that you want to co-localize, you're doing several things. You're adding some noise, and you're also increasing the size over which you are then running the calculations to establish co-localization. So the more black space you have that doesn't have any structure, that doesn't have any of the signal that you're actually interested in quantifying and looking at, then you'll start you'll get wrong values. You'll start to drift away from what the actual relationship is because you're looking at a lot of dead space. So the instances where you'd use the ROI checker here is in those cases where you've got data that's localized to some area, and then maybe some other signal you don't care about, or noise, or just no signal at all. So what Gaurav did is he specified an ROI in Fiji. So we can do that here. We can say Sounds good. So just this one. We can then specify 2 images, the only 2 we have open. We'll use the region from the ROI manager. And in fact, let's do 2 at once. So we've got the analysis for both of those. And wow. From Mander's coefficient, we see a lot of co-occurrence. Values for that are very high, near 1. And for Pearson's curl. Now, this is where we would ask the question of how does the relationship look to us? Does it look to be linear or does it look to be nonlinear? And we can take a look at that on the graph itself. We see it's almost, but not quite linear. Well, yeah, it kind of looks linear. So we definitely want to look at the rank coefficient a little more than we want to look at the Pearson correlation coefficient, which indicates quite a bit of colocalization as well. So that's good. We've got a set of values. Now, let's see what they would be like if we didn't specify an ROI here. What does that change look like? So we can save that log output to reference later. We can, in fact, save all of the data here. This is just a list of all the pixels, all of the values that went into this scatter plot. But let's get rid of these, come back to inputs. None. We no longer care to identify any of the structures. We'll leave the options here the same. We'll click Analyze and let's look at the values there. There is obviously a change in the scatter plot because we're looking at more pixels than before. We're looking at the whole image, not just parts of it. We find what's the PCC? The PCC, quite a bit of change. It went from 0.84 to 0.79, 0.8 almost. There's also a significant change in the Spearman's rank correlation coefficient. And let's look at Mander's. Mander's is close to about the same, actually. And what does that tell us? Well, it tells us that we need to take care when we're deciding what structures to look at and how we look at them, because including a lot of noisy background, a lot of background, or a lot of space anywhere. It doesn't just have to be background. It could be some other signal we're not interested in looking at. The more that we include areas that we aren't really looking for co-localization in, the less accurate our coefficients will describe that data in terms of co-localization. Because these are all, we're looking at correlation, we're looking at we're looking at correlation, we're looking at co-occurrence, and the more we include of area that doesn't have either of those two things, the more we will skew the coefficients in an inaccurate direction. All right, so how is everyone doing right now? Does anyone have questions about what we've gone over, questions about the plugin as well?
Hello, and I want to ask a question. So after the analyzation by this plugin, how can we present our data? For example, in our manuscript, we want to present and to claim that there are co-localization. Just show the SRCC value or PCC value or something like that, or I mean, I just want to know how I can present my result. Thank you.
It would depend on the claim you're trying to make and how you're going about describing it. I would include all of the coefficients you've been calculating. I'd include those in a table, probably along with the scatter plot.
Okay.
As well, and then, you know, any considerations you made in defining your ROI, or just take the reader through what you saw and why you think it indicates colocalization. So the coefficients, definitely the scatter plot.
So maybe the
and
figure
images side by side.
I'll show the table or the data, right?
Yeah.
Okay, thank you.
Let's actually go to that. This guy.
Simple.
Yeah. Because they do it. Actually, I've been looking at those images all the time, but yeah, something like that could be useful, where you've got the data to one side, the scatter plot to the other, and maybe a list of your, like here, a list of the coefficients.
PCC.
Yeah.
Okay. I got it. Thank you.
Yeah.
And one more thing I want to add over there is, and probably you might want to try that out too, when Stoyan compared a cell with the mitochondria versus the entire image with the mitochondria, you can go one more step ahead and draw an ROI only on the mitochondria, right? Because the goal is to know whether the mitochondria in the other channel localizes with the mitochondria in the first channel. And the first channel is mitotracker red, which labels all the mitochondria. So when you play with these images, it would be great if you can try what Stoyan showed to you, those two different comparisons, as well as the third comparison, which is comparing by drawing the ROI on the mitochondria, and then comparing that to see what kind of correlation coefficients you get out of that. So would you like folks to break out and work on the mitochondria examples?
Yeah, yeah, that would be good. So let's take 10 minutes to do that. Is you need to make sure you understand the process. You need to understand the process. You need to understand where the structures might be that you're looking at. And then as you're running the colocalization analysis, you need to keep in mind the strengths of each of the coefficients, and then also the areas where they might be giving conflicting information, as we saw specifically with the Pearson's coefficient. Since it only handles linear relationships well, it could be low for a non-linear relationship, even when the correlation is otherwise very strong. So always keep that in mind, because in a vacuum, it's difficult to interpret these the coefficients numbers, what it actually means. You need the context of the problem you're applying this to in order to use it effectively. Okay, that. Yeah, I think unless anyone has any questions, that would be it for today's talk. As always, feel free to send me or Gaurav any emails with questions. We're always open to consultations. We can schedule a meeting if and when you need one. Otherwise, thank you all for attending.
I also want to credit the authors of the Easy Co-Localization plugin, because they have made a very beautiful plugin which makes it easy to do co-localization. So hats off to them for doing that. And the papers that we sent earlier in the day, they are very helpful to just sit and just peruse through and learn more about co-localization. So thanks to Janelia folks for those papers too.
Definitely, those authors did us a world of service. Well, thank you, everyone. Have a pleasant afternoon.
All right, see you guys next year.